Hi, my name is Samat Satish. My colleague Matan Bonet and I are affiliated with the University of Manchester, and my other colleagues, Professor Sharon Plon and Dr. Deborah Ritter, are affiliated with the Baylor College of Medicine. Our research examined how consistently variant data and clinical presentations are reported on ClinVar, which is a widely used public database of variant assertions, phenotypes, and supporting evidence. We focused on single-gene pathogenic SMARCB1 variants as they are associated with three separate conditions and therefore require clear, consistent reporting of phenotypic presentations, which would eventually help us elicit a relationship between mutation type or genomic position and the associated conditions. Naturally, our research also aimed to identify any obvious relationships between variant characteristics and the three conditions, which include rhabdoid tumor predisposition syndrome, abbreviated to RTPS, schwannomatosis, and Coffin-Siris syndrome. We found 58 single-gene pathogenic SMARCB1 variants available on ClinVar as of January 2023. For each variant, we examined the concordance between conditions asserted by laboratory submissions and those asserted by the cited literature. We also visualized the genomic positions of variants on lollipop plots to identify any obvious patterns of clustering. Only 15 out of 58 variants had both laboratory submissions and cited literature that assert the same condition. The remaining variants were missing one or both forms of evidence or had inconsistent evidence which asserted different conditions. Coffin-Siris syndrome variants consisting of missense and deletions seem clustered in exons 8 and 9. In RTPS, nonsense variants seem to cluster in the 5' end, and large deletions or duplications were always associated with RTPS. Schwannomatosis showed a diverse range of mutation types without an obvious clustering pattern. In light of these findings, and to aid clinicians make accurate diagnoses, we recommend the following. Firstly, given the very different management protocols for each condition, a more standardized way of reporting disease assertions for variants of multiple Mendelian disorders is needed. Secondly, it will be very useful for the submitting laboratory to note whether there is literature evidence or internal laboratory evidence to support assertion of a specific condition. And lastly, more data in the form of patient registries is required for confidently establishing SMARCB1 genotype to phenotype relationships.